Hello everyone, welcome back to our YouTube channel Agri Addict. I'm your mentor Hariprasad SK, a PhD scholar from Indian Agriculture Research Institute, New Delhi. So in the previous video, I discussed about the recombinant DNA technology, which is also called as genetic engineering. So we have discussed about the history and the scientists who gave them, and also we have discussed the methods of DNA or gene cloning, which includes the cell mediated process and also the cell free process. And in today's class, in continuation with whatever we have discussed in the previous class, we are going to discuss the enzymes which are required for the DNA manipulation. So there are certain class of enzymes within this category which is directly involved in the gene cloning, like which is directly used in the gene cloning process, that we will take and we'll try to discuss it in detail. And I'll be also highlighting the things that are important for your exams, so stay with me until the end of the video to get a full benefit. Okay, so let us start with the DNA, start with the enzymes which are required for the DNA manipulation. So that DNA manipulation is nothing but we are trying to just take the DNA or isolate the DNA from one organisms and trying to incorporate it into another, or else we are also the purpose might be quite different, where we want to just amplify the DNA, the gene of interest or a segment of any DNA sequence. So for that we basically go for gene cloning or gene like a DNA cloning, right? So for that we require some enzymes, so we'll look into all those enzymes which are required, and we'll also look into some other enzymes which are actually required for the replication process. Okay, so the enzymes which are required for the DNA manipulation, then they can be broadly categorized into four different types. They can be broadly categorized into four different types. They are starting with, so first one would be DNA polymerase. I hope like you all are very familiar with this enzyme, DNA polymerase. The second one is nucleases, and within nucleases there is a one class of nucleases which are called as a restriction endonuclease, which is actually involved in this DNA cloning process or the gene cloning process. These enzymes are utilized for the successful cloning or incorporation of the target DNA or gene of interest into the host through the vector. Next, there are third class of enzymes, the third category of enzymes, which is called as end modification enzymes. And lastly, we have DNA ligases. Again, the DNA ligases are the very common type of enzymes which we have already learned in various different process in case of molecular genetics. Just we'll be recalling the things that we have already learned in the previous sessions. Okay, so let us start with the DNA polymerase. So DNA polymerase can be either of DNA or RNA. See, whenever we use the DNA polymerase, that means this is an enzyme which is actually involved in the elongation of the DNA strands by adding the nucleotide sequence. Okay, so this is what DNA polymerase. If it is using the DNA and elongating the sequence, that would be DNA polymerase. If it is using the RNA, that would be RNA polymerase. That means if it is synthesizing the DNA strands again, then it is DNA polymerase. If from the DNA strand we are getting the RNAs, which is mRNA, is example is mRNA in case of transcription, it is employed by the enzyme RNA polymerase. So now we are discussing about the DNA polymerase. So based on whether they require template for the elongation of a daughter strand or not. So based on whether they require the template or not for the elongation step, they are categorized into two different types. One is template dependent, in which they obviously require the template for the production of the daughter strands, and in the other one it is template independent. That means without the help of template, without the aid of template, or without the presence of the complementary sequence, they are able to produce the fresh stretch of DNA by adding the nucleotide sequence. Template independent. So these are the two broad categories under DNA polymerase enzymes. So within this, I hope like you are very much familiar with all the different types of template dependent DNA polymerases, because these are the one which we have studied during the replication process. Like for example, we have DNA polymerase 1, 2, 3, and we have several DNA polymerase in case of eukaryotes, and also the other type of polymerase, like we have discussed about the PCR in the previous class, which has this tag polymerase. This is also template dependent. We have seen in the previous class that there should be a stretch of short RNA primers, and then the DNA tag polymerase will come and bind to it, and it can synthesize the new stretch of DNA by using the other one as a template, right? So these are the examples for template dependent DNA polymerase, where the requirement is the template. Only in the presence of the template they are able to produce the daughter strands. So other category is template independent. So the examples for the template independent, there is one important example, which is terminal deoxynucleotidal transferase. So we will discuss its activity in the upcoming slides, but for now just remember this example. It is terminal deoxynucleotidal transferase. So these are the broad categories under DNA polymerase based on whether they require the template or not. The next one. So the next one is the nucleases. So based on whether, okay, firstly we'll discuss what are the functions of these nucleases. What is the role of this nuclease? What does these enzymes do? So the nucleases are basically enzymes which actually digest the fragment, like digest the sequence of either DNA, they digest the sequences, and the sequences might be either RNA or DNA into the fragments. That means they digest any sequence of either RNA or DNA and produce a number of fragments which will be eventually degraded. Or it can be also said that these are the enzymes which will degrade the DNA or the RNA sequences. Based on whether they degrade the RNA or DNA, they can be called as RNase enzyme or DNase enzyme. DNase enzyme is a nuclease which will digest the DNA, and RNase is an enzyme which will digest the RNA. This is one type of category. And next, based on their location of action, they are divided into two different categories. So what are the two different categories? They are called as exonuclease and endonuclease. So these are the two important categories under nucleases, which is based on their location of action. So for example, like this exonuclease, the name itself suggests it makes the cuts, or it digests the DNA sequences from the ends. For example, it is double-stranded DNA from 5' to 3', in opposite it is 3' to 5'. So the exonucleases, what they will do, they will just degrade the nucleotides from the end one by one. So this is the action of exonucleases, and the exonucleases can act either from the 3' direction or from the 5' directions, and there are also some of the exonucleases which can act on both the directions. Okay, all the categories are available which can act on a DNA, but the difference is they can act only at the free ends, like they will be acting at the ends, and they can degrade or remove or break the bonds of, like break the phosphodiester bonds from the ends of the DNA sequence. So this is about exonuclease. Next we have this endonuclease. Endo, from the Greek term, it is means within. Endo means within. That means it will produce the internal cuts. The endonuclease, it will degrade the DNA by producing the internal cuts. That means it can act on any stretch of DNA at any location, not at the ends. Like, okay, it can also act in the in between the sequences, in between the nucleotides also. So these are called as endonucleases. I hope like these two differences are clear. There are several examples of the exo and the endonucleases. Like for the endo we have mung bean nuclease, S1 nuclease, and so many. Okay, so you can remember either, like you can just remember at least 2 or 3 different examples which are important, which you feel that are important. Okay, within this endonucleases, again we have a subcategory. The endonucleases are the one which degrade the DNA or which makes the internal cuts. Within this endonucleases, we have a certain class called as restriction endonucleases. I hope like you might be very much familiar with the term restriction endonucleases, since it has a wide application in the field of genetics and plant breeding, and also in the biotechnology. You might be already knowing this term restriction endonuclease. Let us just know in detail about these enzymes, and if there are any classification or categories, we will discuss all of them one by one. So restriction endonucleases. See, what are these basically? These are again a class under the endonucleases. Obviously, we know the function of endonuclease. What is it? It means it may degrades the DNA by producing the internal cuts. But what is the speciality of this restriction endonucleases? See, the speciality or the important features of this restriction endonucleases are, firstly, they recognize a specific sequence on a DNA strand. That means a specific restriction endonuclease will identify or recognize a certain stretch of, for example, it is ATGC, TAGC, AG, something like that. If I just randomly place it, so every restriction endonucleases has a specific recognition sequence. What I mean to say is it can recognize only a specific sequence. For example, I have one restriction endonuclease enzyme, it is echo R1. If we assume hypothetically that it will recognize certain sequence called as ATGC CA. So this is a sequence and that it will be recognizing, and it is recognizing 6 base pair sequence. If it is recognizing 6 base pair, it is called as hexacutter. So this is a enzymes, just for example. Yes, it is a obviously hexacutter, but the sequence what I am showing here is it's just arbitrary. Okay, so this echo R1, it is a hexacutter. That means it will recognize a specific 6 base pair sequence on a DNA strand. That means if I provide this restriction enzymes to this DNA sequence, if it has this sequence within itself, then it will create or it will make the internal cuts exactly at these points. Okay, and both the strands on the both the strands, it will exactly make the cuts on this stretch of DNA which is specific sequence. So because of that reason, they are called as restriction endonuclease. That means they will recognize certain sequences and they can make only cut at that particular sequence. If in case a certain stretch of DNA to which this endonuclease is restriction endonuclease is introduced, if the stretch of DNA doesn't contain the specific base pair sequence that the enzyme recognize, then there won't be any cuts. That means there won't be degradation of that DNA by that endonuclease enzyme. Only if there is a specific restriction site or the recognition sites that is available for that enzyme, only then it can make a cuts and degrade it. Okay, I hope this is clear. I'll tell you like why the name restriction. Okay, the name what we discussed, it is restriction endonuclease. I told you the special feature of this restriction endonuclease is it will recognize a specific base sequences on a DNA, where the different endonucleases can recognize different set or different unique base sequence. Either it might be a 4 base pair sequence or a 6 base pair or a 5 base pair or any length. Okay, and the one which I discussed, it was echo R1. It was an enzyme which recognized the 6 base pair sequence. So before I talk about this restriction endonucleases in detail, let us know about it history. So it was for the first time by W. Arbor, who postulated the presence of this restriction endonuclease. Next, after that, there were two scientists who were Smith and Nathans. Smith and Nathans were the first scientists to isolate a restriction endonuclease which was named as IND2. So IND2 was derived from a bacteria which was Haemophilus influenzae. Haemophilus influenzae. Just remember the name. This is H for Haemophilus, IN for influenza, and we'll also discuss like how this actually name is derived in detail in the upcoming slides, but for now just know these things. Now, so why the name restriction for this endonucleases? The name endonucleases, the meaning we already know it, why it is called as endonucleases, but why the name restriction endonucleases? So basically these enzymes which we are talking about, the endonuclease, these are produced in which organisms? These are basically produced in a bacteria, in the bacterial cell. So these are produced. So what is the functions of this endonucleases? It is to protect the bacteria. So these are actually produced to protect the bacteria from the foreign invasion. That means bacteria from the foreign invasion. That means if there is any foreign DNA that is coming into the bacterial cell, these endonucleases will get activated and they will degrade the incoming DNA, or they will restrict the movement of the external or the foreign DNA into the host cell. So that is why the name restriction endonuclease. That means these set of endonucleases will restrict the movement or the entry of the foreign DNA into the host cell. The host cell is bacteria here. So that is why the name is derived as restriction endonuclease. I hope this is very much clear to all of you. Next, after in D2, there was a second enzyme which was isolated. It was echo R1. So the name echo R1 is derived from Escherichia CO4 coli from a strain RY with which is numbered as 1 based on its discovery. Okay. The strain name is RY13. E. coli RY13, and the 1 refers to, like, the Roman letter 1 refers to the order of discovery. This was the first enzyme that was discovered in this particular strain, so that is why the name or the letter or the number, like Roman number, which is 1. So this is about echo R1. So now let us just talk about this nomenclature, how these are named. So this nomenclature might be important for your exam point of view. Just make sure to remember this. This is very simple. See, there are actually 3 parts. You can see, like, this is the first part, and R is second part, and 1 is third part. First part, second part, and third part. And the first part may include the name of the genus, 2 to 3 letters of the genus or the species that is included here, where E refers to Escherichia genus and CO refers to coli. And the second part is derived from the name of the strain that from which it is derived. And the third, I told you, it's from the order of discovery from its particular strain. And in the second part, it can be a number or a letter or combination of both. Okay, so these are the 3 different parts which are used for naming the restriction endonuclease. So try to remember these points. Echo R1, here the echo means Escherichia coli, and R is a strain, and 1 is order of discovery, and this is the hexacutter, and the other features we'll discuss in the upcoming slides. Next, I told you about one important features of the restriction endonuclease, which is that it recognizes the specific base pair, specific bases on the DNA strand, right? So this was the one feature, and the next feature is they provide some specific cuts, and the types of cuts that are produced by the restriction enzymes, they are classified into 2 different types. One is blunt ends, that the restriction enzymes can digest the DNA and create the blunt ends, or it can produce the staggered ends. Staggered ends or stagger cuts, stagger ends or cuts, or blunt ends or cuts, both are same. And staggered cuts, it is also called as sticky ends or sticky cuts. So what is the difference between these two? So if you just clearly observe, this is the echo R1, which is, I told you, it's a hexacutter, though it recognizes a specific 6 nucleotide sequence. Here the specific 6 nucleotide sequence that is recognized by the echo R1 is GAATTC. So in this, echo R1 produces a sticky ends. What does it mean? So this is a complete restriction site or the recognition site that this enzyme is recognizing. After recognizing this particular length of DNA, it will make a cut in such a way that it would be like a sticky end. That means it will have some overhang nucleotide sequence in one of the 2 strands. For example, if it is a 5' to 3', and it is 3' to 5'. And here you can clearly see in the one strand there is a overhang sequence that is blank in the first strand, and in the opposite strand, in the complementary strand, in the opposite direction, there is a same staggered cut or the same overhanging sequence. That means there are blank. And the similarly, if you just see in the other one which produces the blunt ends, this is by the echo R5, so it creates the blunt ends. So again it is a hexacutter, and the specific nucleotide sequence that it is recognizing is GATATC. So this is a specific nucleotide sequence it is recognizing. But if you look at the cuts, there is a one horizontal, like one vertical cut, the straight vertical cut that has been made, and you can clearly say these two are divided into equal parts. There is no stacked cut here. There is no formation of sticky ends. There is a blunt end formation in the second case. So that is what the blunt and sticky ends means. We'll discuss like what is the need or what is the application of these sticky ends in the upcoming slides, but for now at least know what are the sticky ends. Sticky ends are nothing but the production of overhang structures on both the strands. That means at some nucleotide sequence there will be only single stranded, not the double stranded. Only single stranded sequence will be present, and there will be a certain gap between the both the strands. So that is what sticky ends means. Next, we'll discuss about the features of the restriction endonuclease. So what are the features? I think we have discussed about two features already, which are the production of either the stacked ends or the sticky ends, stacked cuts, or the blunt ends or the blunt cuts, right? So we have discussed about this point, and the previous to that we have discussed about the recognition of specific nucleotide sequence. That means it will recognize a fixed number of, like it will recognize a fixed nucleotide sequence and produce the fixed number of fragments. That means if this is a stretch of DNA, if there is a specific nucleotide sequence that is repeated two different times here in the stretch of DNA, if I introduce the restriction endonuclease to this particular DNA or a media containing this particular DNA, then it will cut exactly at these two positions. So it will make a fixed number of fragments by identifying the restriction site or the recognition site. So this is the one important feature that we have already discussed. Next important feature of this restriction endonuclease is, see, whenever they produce the sticky ends or whenever they make the staggered cut, you can clearly see that I told you in the previous slide, it makes the cuts such that there will be a single stranded region in the both the strands. But if you closely look at the strands, they are palindromic in nature. That means on both the directions, if you see, they are the same nucleotides, right? On both the strands, they are the same nucleotides when they are read in the opposite direction. GAATTC, in the opposite direction it is again GAATTC. So this is the nature of the unique restriction sites that are identified by the endonuclease. So here why I use the term sticky or staggered ends, because the diagram shows the formation of sticky or staggered ends. But the palindromic sequence, these are the general features of the restriction endonucleases. That means the unique restriction sites that the enzymes will recognize, that unique sequence will be palindromic in nature. That means in both the directions they are read as same nucleotides. Next feature is the production of the stacked cuts or the blunt ends, which I told you in the previous slide. See, these are the important features of the restriction endonucleases. One is unique restriction sites, second one is palindromic sequence, third one is production of either staggered cuts or the blunt cuts, blunt ends or cuts. Okay. Next, there is a classification of endonucleases based on site of the cut which are made and the sequence that are recognized, and the ends that are produced. So based on that, these restriction endonucleases are categorized into three different types. One is isocysiomers, and second one is called as neocysiomers, and last one it is called as isocodamers. Okay, so this is very much simple to understand. There is nothing much deeper or nothing much conceptual things. So that's why I'm trying to explain it, because I cannot exactly say whether this will be asked in the exams or not. There is always a probability that these type of questions can be asked. So I'm just trying to explain these things. So since it is very much easy to understand, it doesn't require much time to take up. Okay. So first one, the isocytosomers. So under this category, there are different enzymes, like different enzymes, they produce or they recognize same sequence. That means different restriction endonuclease enzymes. There are two different restriction endonuclease enzymes, but they recognize same sequence, and also they make the same cuts. So this category of the different endonucleases are under isocytosomers. That means different enzymes, same location, like same sequence of identification and same location of cuts. This is under isocytosomers. Second one, it's neocytosomers. Again, there are different enzymes and same sequence identification, the same restriction sites, but different cuts. The cuts are made in the different location. That means if this is a certain stretch of DNA ATGC TA, and in the opposite strand TACGAT, then if I introduced two different DNA, for example, two different restriction enzymes 1 and 2, since both have same restriction site, that means recognition site or restriction site, that means they identify the same nucleotide sequence, they can cleave this DNA. Both of them can cleave the same DNA and at a same sequence, but at a different location. For example, one is cleaving at this location, the other might be cleaving at some other location within the same restriction site. So that is what neocytomer means. Lastly, we have the isocodamers. So what are these isocodamers? So these are different enzymes with a different restriction site. Again, this is also different here. And next, different location of cuts that are made within the restriction site, but the ends that are formed after the cuts are same. The ends that are made after the cuts are same, but they are the different enzymes that recognize the different restriction sites, and the cuts are produced at a different location, but the final ends that are made by the different restriction enzymes, they are same. So these are under isocodamers. And there are certain examples, like for example, for isocytosomers, the examples are SHP1 and BBU1. And for neocytosomers, the example are the SMA1 and XMA1. And lastly, for isocodamers, the examples are SAM3A and it is BAMH1. So these are the examples. You may not require to remember the examples for the ICR PG exams, but at least know about these terms and their meaning. So that would be important for you, might be asked in the exam. Since it is very simple to understand, I hope like it doesn't cause any difficulty for you to know this. Next is the calculation of the frequency of the restriction sites. So this is very much important for your exam point of view, and it has been already asked in ICR PG exam 2023 in the shift 2 paper. So this was a question that was there in the shift 2 paper. I have taken the question, and while solving this question I will explain the concept. So the question says, on an average, how many fragments would a restriction enzyme which recognizes a specific 5 base pair sequence in a DNA be expected to cleave the double-stranded bacteriophage with a genome of 6066 base pairs into? So what they are asking is, they are asking like how many fragments a certain restriction enzyme which recognizes this 5 base pair sequence would be expected to cleave. Like how many fragments can a certain restriction enzymes can provide if the stretch of DNA, the length of the DNA is 6666 base pairs. That means the total length of the DNA which we have is 6666, and we have certain restriction enzyme which can recognize a 5 base pair sequence. It might be anything, 5 base pair sequence, and it can make cuts wherever it gets to see this unique 5 base pair sequence. That means, like, if for example AT GC AT, sorry, A, so these are the 5 base sequences that you can see. And for example, let us assume that the restriction enzymes can recognize these 5 base sequences, and wherever these sequences are made, it can form the cuts. So this is what they are asking. So if this is the case, if, like, in how many copies this particular unique sequence is present, that many cuts will be produced. If this particular sequence is present in the multiple copies in a genome within this 6666 base pairs, that many cuts will be produced. But we have to first calculate, like, how many times this specific base sequence is repeated within the 6666 base pair. That means the total length of DNA is 6066 base pairs. Okay, within that 6066 base pair, we have to calculate or know, like, how many times a specific 5 base pair sequence is repeated. Okay, so this is what the question is saying, and this is our understanding about the question. So let us see, like, how to solve this. What int they have given? They have said that it will recognize the 5 base pair sequence. Okay, so this is the first one. Okay, how many number of nitrogen bases are present? We know it's 4. It would be ATGC in case of DNA, and it would be AUGC in case of RNA. Now, since they have asked for DNA, we'll consider the same. And even if they are asking for RNA, the procedure and everything is same. Need not to confuse with the things, because the RNA will also have the 4 nitrogen bases, and the DNA will also have the 4 nitrogen bases. To make it very simple, what I'm trying to say is, to make you understand, let me give some example. Consider there are 4 different colored balls: black color ball, red color ball, and brown color ball, and another one is green color ball. So these are the 4 different color balls I have, but the number of balls, the total number of the balls are unlimited. I have unlimited number of balls, but the colors I have is only 4 different colors, but the number of balls are unlimited. Okay, so if I ask you to arrange these balls in an order of in the 5 blank spaces such that the arrangement should not be repeated again. That means, in the first case, if I place all the black balls in all the places in the 5 arrangement, this combination should not repeat again. With like, excluding the first combination, what are the other combinations that I can make? I can place the first black ball here and make others red, and in the third I can place the black ball in the 2 places, and rest I can place it with the other color. So likewise, how many unique number of combinations I can make using these 4 color combinations? How many unique number of 5 row length I can make with the use of these 4 different colors? So how to calculate it? If you remember from the 11th and 12th mathematics, it's very simple. It is just 4 power n. 4 is nothing but the number of colored balls. It is, since we have 4, so this is 4. What is n here? N is number of blank spaces, or how many blank spaces we have? We have 5. That means, how many unique combinations that we can make? We can make up to 1024 unique combinations. Okay, there are total of 1024 unique combination we can make. Similarly, just replace these balls with the nitrogen bases now. That means it is adenine, guanine, and cytosine, and lastly thymine. So these are the 4 different nitrogen bases I have, and it should be arranged in the 5 blank spaces side by side, and it should be unique. That means the specific sequence should not be repeated again. Then how many unique combinations I can make? Again, it is 1024. So this is the unique combination again make. But what is the total stretch of DNA I have? I have around 6066. If I divide this 6066 with 1024, then I can then I will get number of times the unique combination is repeated. That means this is out of 1024, there will be only one unique combination of all the possibilities in the 1024, but in the 6066 how many will be there? That can be done by just dividing it. It will be approximately 5.9. That leads to approximately 6 base pairs. So that is why they have also mentioned that it is about, not exactly, it is about 6 base pairs. That means it will make us 6 fragments when we use the restriction enzymes, which can recognize a specific 5 base pair sequence or base sequence with when the length is about 6066 base pair. I hope this is very much clear to all of you. If you have any doubts, you can comment on the video. I will try to explain it again. Next, so there might be one question for you now. That is, like I told you earlier, that these are the enzymes which are recognized by the bacteria, which will degrade any foreign DNA that is being inserted into, or that is being entered into the bacteria. They will recognize the foreign DNA if this is the foreign DNA, and they will degrade it. But the question arises here. What is the question? How the restriction enzymes is able to identify or recognize the certain stretch of DNA as foreign DNA? How it is able to, for example, this is a nucleoid and within that genetic material is present and the proteins are produced by this, and for example these are the restriction enzymes which are produced. This restriction enzymes, how it will know that this is a foreign DNA and this is not the foreign DNA and this is the bacterial DNA? How it will differentiate between the foreign DNA and its own DNA? So this is the thing, right? If it couldn't recognize or differentiate between the foreign DNA and its own DNA, then it should also cleave its own DNA or degrade its own DNA. But it only cleaves the foreign DNA. How it will recognize this foreign DNA? This is by modifying the own DNA. So the own DNA of bacteria is modified by methylation process. So methylation usually occurs in cytosine residues or the adenine residues, and this will undergo methylation just like a marking, so that these methylations can be recognized by the restriction enzymes. Wherever the methylations are present, that DNA, it will consider that its own DNA and they won't degrade it. Okay, so this is about recognizing itself and differentiating between the foreign DNA and its own DNA. And under this we have this restriction modification system. This is why, like, see, the restriction modification system is certain system which is actually required to differentiate the foreign DNA and its own DNA. For this system is required. Okay, and this restriction modification system, if we break this is a restriction system and the modification system. So restriction system, what is the function of this restriction system? To digest the DNA. Then what is the function of the modification system? To modify its own DNA. Modify its own DNA. That means, I told you earlier, it will mark its own DNA by how it will modify. It will methylate its own DNA at which residues? It is either cytosine or adenine residues. Okay, so this is how the modification occurs, and this system is called as restriction modification system. So based on this restriction modification system, and also based on the enzyme complexity, cofactor requirement, enzyme complexity, the cofactor requirement, and the position of cleavage, where exactly the cleavage is occurring. Based on this, there are 3 different types of this restriction modification system. So they are called as type 1, type 2 and type 3. Again, this is the name which you might be already very much familiar with, the type 2 restriction enzymes, type 3 restriction enzymes. So these are the categories, and basis of which they are categorized based on the enzyme complexity, whether they have, for example, in the first case, that means whether they have both restriction and modification function, or it has only the restriction function. So this is what the complexity refers to, the enzyme complexity, the functions of the enzymes, whether the single enzymes can act on both restriction and modifying. That means both can either act as degrading the DNA and also modifying DNA or not. Based on that, they are categorized into three different parts, three different types. So first one, that is first type 1, type 2 and type 3. Out of this the three different types, just remember that 1 and 3 are ATP dependent. That means for their action to take place, they need the energy in the form of ATP, and these have the three different subunits, and type 2 has two identical subunits, and type 3 have two different subunits. Next, the nature of enzymes. See, the single enzyme in the type 1 and type 3, the thing is, the difference is, these type 1 and type 3, the single enzyme can act as both restriction or restriction digestion and modification of the genome. So it can act both as a restriction digestion and also modifying the DNA, both type 1 and type 3. But in the type 2, there are different enzymes. That means a single enzyme can only perform a single function. A single enzyme either can perform the endonuclease function, or an enzyme can either function the methylase activity. That means there are two different enzymes for the two different activities, in simple words. Next, the cleavage site. So if you look at the cleavage site, in the first case, so this is by, see, the cleavage site of type 1 and type 3 are bit similar, and type 2 is unique. How? In this case, for example, this is a restriction sites or the recognition site that the endonuclease can recognize. Okay, but even after recognizing this specific site, it won't provides the cuts within this sequence. It will provide the cut very far away from the recognition site. So this is in case of type 1, approximately around 1000 base pairs, up to 1000 base pairs away, it will create the cuts. This is a type 1. And in the type 3, you can see it will create the cuts 24 to 26 base pairs 3' to the restriction sites. That means it will create the cut 26 to 24 base pairs at the 3' end, here and in this side. This is where the type 3 will make the cuts. Type 1 and type 2, the similarity is that they won't make the cuts within the restriction sites. They make the cuts away from the restriction sites. In the type 1, it is far away. That means up to 1000 base pairs away. And in the case of type 3, it is up to 25 to 26 base pairs away in the 3' site. That is also very much important. But in the type 2 restriction enzymes, they recognize a certain sequence and they make the cuts within that recognition site. You can see clearly the arrow mark signifies that within the restriction site the cuts have been made by the type 2 enzymes. But the other two different types, like type 1 and type 3, they can make the cut away from the restriction site. Okay, so for this reason, why I'm trying to emphasize this is, for this particular property of type 2, which can cut within the restriction sites, this is primarily used in case of DNA or gene cloning. DNA cloning or gene cloning. The type 1 and type 3 cannot be used for gene cloning for this purpose. Why? Because in this, like, the cuts which are made further away, they are not same at every time. For example, if there is a one stretch of DNA, I introduce the type 1 endonuclease. In this type, if it has made a cut at certain location, in the second time, if I introduce the same endonuclease to different stretch of DNA, then it won't be producing the same cuts at the same location. This location will differ from the sequence to sequence. Because of that, we cannot use it in the gene cloning or DNA cloning. Why? Because in the DNA cloning or gene cloning, we have to make the cuts at a same location, both on the target gene or a target DNA and also in the vector which we are using as a vehicle, which will be mostly the plasmid or the viral genome. So cut should be exactly at a same position in both vector and also in the target DNA. Only then these both can be joined and combined to form a recombinant DNA, which we discussed in the previous class. Okay, so for that reason only type 2 is useful for us. So let us understand this recombination event once again. This was under recombinant DNA technology, which we discussed in the previous class. See, if I have two different sets of DNAs from two different species, like DNA from species 1, DNA from species 2, what I have to do if I want to recombine them? I have to cleave them using a same restriction enzymes. Here in the figure they are using echo R1. So the echo R1 sites are present in both the species of DNA. Then I can use this particular restriction enzyme. After using this restriction enzyme, as you can clearly see, in both the species it is creating the cuts at exactly the same position. There is not at all a single nucleotide difference here. It is exactly making a cuts at the same position. Because of these cuts, as I told you, the echo R1 produces the staggered cut, or the produces the sticky ends. You can see that there is a overhang structures that are being produced in both the side. That means the location of cuts are different on both the strand, because of which we produce the we get the sticky ends. And this, because of the production of these sticky ends, this particular strand can match with the strand of the different species. And you can clearly see by the change in the color that this is a DNA from the species 2 and this is the DNA from the species 1, which were able to easily ligate with each other because they have cut by using the same restriction enzymes, which is of type 2. And this is the recombinant DNA that we have received at the end, recombinant DNA molecules. I hope this is very much clear. So we have discussed about the nucleases. Nucleases are two different types, the exonuclease and the endonuclease. And the endonuclease, we have again a subtype which is a restriction endonuclease. Why the name restriction endonuclease? Because it will restrict the entry of the foreign DNA into the bacteria, and these have a special properties which are they are palindromic in nature. That means they recognize the palindromic sequence and they recognize a specific number of unique sequence, and they produce either staggered cuts or the blunt ends. Blunt cuts or staggered cuts, staggered ends or the blunt ends. So these are the important features of restriction endonuclease. And there are again of three different types, that is isocysiomers, neocysiomers and isocodamers. And based on the enzyme complexity, cofactor requirement, they are divided into type 1, type 2 and type 3, within which the type 2 is actually used in the DNA cloning or gene cloning because it makes the cuts within the restriction sites, and the other two makes the cuts far away from the restriction sites. I hope like all of these things are clear. You can expect any type of question within this, so make sure to understand the concepts very clearly. I hope like it is also very easy to understand. Next one, so third category of the enzymes which I told you is end modification enzymes. So this is also very simple to understand. See, first enzyme is terminal deoxynucleotide transferase, TDT, and second one is alkaline phosphatase, and the third one is T4 polynucleotide kinase. So these have different functions, but all of them are having a similar role. That means they modify the ends of the DNA sequence. That is the common thing between these three different enzymes. First one, the terminal deoxynucleotide transferase. If you remember from the previous slides, I told you this is the example for template independent polymerase. It doesn't require the template for the synthesis of a nucleotide sequence. So this is terminal deoxynucleotide transferase. So what it will do, it will add the homopolymers. It is involved in homopolymer tailing. If this is a stretch of DNA 5 prime to 3 prime and 3 prime to 5 prime, so this will add homopolymer, like single type of nucleotide, either A, either T, or either C or G, into the C prime end, with the help of this enzyme. Without having any template, it will still add the nucleotide sequence. That is why this is template independent, and this is at the both the ends. Like at the 3' end, it will add a single type of nucleotides. Okay, so this is TDT. Next we have alkaline phosphatase. So this alkaline phosphatase, this enzyme will remove the phosphate group that is present at the ends of the chromosome, like ends of the DNA sequence. It will remove the phosphate groups from the ends of the chromosome. This is how they modify it. So what is the like consequence of removing the ends of this ends, like removing the phosphate group from the ends? If the ends are like phosphate group from the ends are removed, if there are two different DNA sequences, this is one and this is two, and from both of them, like the phosphate group from the ends are removed, then even if I use the DNA ligase, I cannot join these two, because phosphate group is very much required to form the phosphodiester bond. If I use this enzyme, then this ligation process can be stopped. So this is the like consequence of using the alkaline phosphatase. Next we have a T4 polynucleotide kinase, which is, see, this is terminal deoxynucleotide transferase. Here you can see that single type of nucleotide is being added at both the ends, at the 3' ends, in the both the direction, in the both the strands, they are being added. So this was about terminal deoxynucleotide transferase. And the second one, the alkaline phosphatase. I hope like you might be knowing at at both the 5' and on the both the strands there will be a phosphate group, and this will remove this phosphate group in both the ends, both the 5' ends. Next we have the third one, the polynucleotidal transferase, T4 polynucleotidal kinase, sorry, T4 polynucleotidal transferase. So this T4 polynucleotide transferase, this is having a opposite action of the alkaline phosphatase. That means it act as addition of phosphate group. So this function is to add the phosphate group. In the first case, it is, there are two different types of reaction here. One is called as forward reaction and other one is called as exchange reaction. The forward reaction is, there is certain stretch of nucleotide sequence that doesn't have this phosphate group at both the ends. Like in the 5' end there is no phosphate group. Obviously it should be there in the 5' end, but there is no phosphate group. With the help of this enzyme, the introduction of this enzyme will add the phosphate group to the 5' end. So this reaction is called as forward reaction. There can be other type of reaction. If for example there is already a phosphate group which is present within the DNA sequence, then there is a exchange reaction in which first this phosphate group is removed with the help of conversion of ADP to ATP, which takes place, and then utilization of ATP again, the new phosphate group is added to the same DNA sequence. That means the older one is removed and the newer one is added. So that is what exchange reaction means. And where it is utilized, this is utilized in DNA labeling. If I want to label certain stretch of DNA, then I can go for this addition of the phosphate groups, the labeled phosphate groups, with the help of T4 polynucleotide transferase. This is the application of it. I hope this is very much clear to you all. Next, so the last category of the enzymes is the DNA ligase. So the DNA ligase, what is its functions? The DNA ligase joins the ends of the two different strands. But if the ends of the DNA are sticky ends, if the ends of two different stretch of DNAs have sticky ends, then there is a higher efficiency in the DNA ligation. That is because of presence of the complementary sequence. See, this sequence will match this sequence so that they can be easily recombined, and there can be higher efficiency of the DNA ligation. But if the blunt ends, the efficiency is less when it is compared with the sticky ends. So this is about DNA ligase, and almost all the DNA ligase which are used for all this purpose, they are derived from the E. coli or from T4 bacteriophage. Okay, and what does this DNA ligase will do? They will add phosphodiester bond. So this is very much important. So how they will link the two different strands? They will add the phosphodiester bond between them, between the two different strands. This is how they will be able to ligate or join the two different strands of DNA. Okay, linkers and adapters, I will this is not as such important. If it is required, I will make a separate video on that. This is very simple to understand, and then I will upload in the YouTube. For now, you can drop it. So this is what I wanted to discuss in today's class. If you have any doubts regarding the things that I have discussed in the previous video and this video, feel free to contact me, or you can comment on the video. We'll surely answer your questions, whatever it is. Okay, and if you are looking for a complete mock test bundle for your upcoming ICR PG exams, which will be conducted in another 2 days, you can register our online mock test bundle, which is available for just 149 rupees for now, which contains full topic wise mock test, full length mock test, subject wise mock test, which will be helping you to better evaluate yourself and do your best in your exam. Okay, that's all for today's class. If you like our content, please like, comment, share and subscribe our YouTube channel. Thank you.